So, good morning, or good afternoon, or good evening, wherever you're joining us from. A warm welcome to today's webinar, organised by BMJ in partnership with the Asia Development Bank. I'm Dr. Kieran Walsh, Clinical Director at BMJ. I'll be chairing today's webinar, which is focused on COVID-19 vaccine safety from bench to bedside. We are delighted to be joined by an international panel of speakers who are going to share their knowledge and expertise in this area. We'll be ending the session with an audience Q&A, so please do submit your questions via the Q&A button. If you have a comment or would like to tell us where you're joining, you can use the chat box. Please select all participants from the drop-down menu when using the chat box. Before we begin, I'd like to let you know that all microphones and videos have been disabled for this webinar, and the session is being recorded. Throughout the webinar, we also have a few polls to gather your feedback. We have people joining us from many different countries, so it'll be interesting to see what your collective responses are. Please do take part. So let's start with our first quick poll to hear some of your thoughts on this topic. So Mark, if you can please launch poll 1. And so Mark, if you can show us the results from poll 1, please, that would be great. So interesting, a real mix of response, but actually, if you see yes, yes, widespread, yes, only in some populations, there definitely is vaccine hesitancy. So I'd now like to introduce Dr. Patrick Osewe, Chief of the Health Sector Group at the Asian Development Bank. Dr. Osewe provides leadership on policy, technical and operational matters and leads the application of evidence-based and innovative approaches to address priority health issues in Asia and the Pacific. You're welcome, Patrick.
For the introduction. So the Asian Development Bank is the second largest development bank in the world. And we've been providing support and financing for Asia for over half a century. In those years, for almost 60 years, Asia has never had any severe recession. And COVID-19 is basically reversing the gains that Asia has made over the past 6 decades. And so this is something that we take very seriously. And as such, we've invested very heavily to support our developing member countries. And I'll just summarize our support is in 3 ways. First is convening of partners. And I'll give an example whereby last year we convened the ministers of health and ministers of finance. This was the first meeting in our region ever to bring together ministers of health and ministers of finance to talk about a health issue. And the focus there was to see what is it that can be done jointly between the ministers of finance and ministers of health to accelerate progress towards UHC as a way of ensuring that if any pandemic that starts can be picked up at a very early stage. Second, we were the Asia and the Pacific. The economy is driven in many countries by tourism. And so we brought together leading experts around the world to discuss how to reopen the tourism sector. So for example, as you travel, when you leave your home to go to the airport, the first interface is the airport authority that you meet. And the staff of the airport authority is owned in many countries by the Ministry of Transport. The second, when you check in, you meet representative of the airline. This is a private sector company that you meet when you're flying. Third, you go to immigration, and immigration falls under the Ministry of Home Affairs. And when you arrive and you go through customs, that falls under the Ministry of Finance. And then you go to your hotel, which is privately owned. And if you're a tourist, you go for tours, to a company. And then, of course, the airline is made by also a private sector company like Boeing or Airbus. So to bring all these people together to discuss how to address the issue of safety when you're traveling was a mammoth undertaking, but we were able to come up with very good recommendations in that regard. Second, we do a lot of knowledge generation. For example, for currently, we are involved in many opportunities to talk about pharmacovigilance, and this is a good example of that. Risk communication, generating knowledge on how is the best way to communicate the issues of risks and the issues of, for example, what was on the question about hesitancy. Capacity building, which is required because COVID-19 was basically a new disease that not many people had experience on that. The issues of logistic management for vaccines and commodities, big issues. The issues of coordination. And right now, we are very much involved in exploring options for how to build the capacity of countries in manufacturing. Then the last is financing, which is extremely important, but you need to be able to know exactly what you're doing and what you're going to do with the money. So financing, we provided $20 billion initially for a COVID response, and then recently in December last year, we allocated $9 billion to Asia and the Pacific, which is the highest financing for vaccine per capita worldwide. So I'm very much happy to be here, and our partnership with the BMJ is to build the capacity and to share knowledge by bringing experts together. So I would like to welcome our esteemed experts today to this meeting. Over to you, Kieran.
Thanks very much, Patrick. Now, I'm delighted to introduce Dr. Stephen Pryor. Dr. Pryor is a PhD microbiologist and the president of Therax. Dr. Pryor has over 35 years of experience in working with both public and private sectors to provide policy and technology solutions for issues impacting national security and health. His work includes the focused growth of biotechnology and pharmaceutical projects, from initial research to licensed products, including diagnostics, therapeutics, and vaccines. He's also led the development and implementation of policies, procedures, and legal instruments for public health responses to biological threats in the United States and internationally. Dr. Pryor will be speaking about vaccine clinical trial safety. Dr. Pryor.
Thank you very much for that introduction. That's fine, Kieran. And thank you to the BMJ and the Asia Development Bank for putting this session on, which is a very timely session, given where we are now with dealing with the use of the vaccines. And based on that poll that was put up earlier, some of the hesitancy that exists around the safety and efficacy of these vaccines and hesitancy to use them. So I'm conscious of time. I'm going to give a short presentation and recognize that my colleagues have time as well. So everything that's on my slides is referenced, and to the degree that anyone wants to follow up, they can follow up with me or via BMJ, but I am going to go through the slides, and the slides will have references on them. So let's go ahead and do that. Okay, is that clearly visible?
Yes,
okay.
it is.
Yes, it is.
Thank you. All right, so I'm going to proceed. As Kieran said, I'm going to talk a little bit about Sars CoV-2, the virus, the disease COVID-19, but I'm going to focus on the issue of the clinical trials and vaccine safety that have preceded the use of those vaccines globally. The topics I'm going to cover are a little bit about vaccination, which I think is important to put it in the context of vaccine safety and efficacy. I'm going to do a comparison of the vaccine development process to give some ideas about why globally we were able to achieve the phenomenal success of obtaining vaccines, launching vaccines, and using vaccines against this disease in an unprecedented time frame versus normal vaccine development. But the bulk of the time I'm going to focus on the COVID-19 vaccine clinical trials. And obviously in 15 minutes, I can't cover much, but I'm going to take one example and explain how vaccine trials are conducted and the results from them, and then leave it to my fellow panelists to discuss with you what happens after clinical trials when we move into the post-vaccine use monitoring. So I'd like to start by just identifying that this is an unprecedented environment in which we've been developing vaccines. And somebody phrased it as building the plane when we fly. It's clear that developing any vaccine or treatment in the middle of a pandemic is never ideal, and it presents many, many challenges, and many of those have been well documented. The good news as far as Sars CoV-2 and COVID-19 is concerned is that this is not our first vaccine, and it won't be our last either. So it's not that as far as COVID-19 and SARS is concerned, this was our first vaccine. It was a series of vaccines that have been developed over time, and our vaccines that we're now able to take advantage of use vaccine technologies and platforms that are adaptable from other diseases. So we start from the premise of needing vaccine capability, and we then adapt those in order to address the issue of Sars CoV-2 and COVID-19. It's also important to note that prior coronavirus disease and vaccines has provided key information related to this current pandemic and outbreak. So the work that was previously done on SARS, the original virus associated with the SARS disease, MERS, the Middle East Respiratory Syndrome, and also some of the early work that was done on human coronaviruses back in the late 80s, when I was still doing research, have actually been quite helpful in telling us some of the things we needed to know about COVID, including looking at the immune response, protective immune response, what we were looking for in a vaccine. The other good news is that the pandemic concerns related to COVID-19 facilitated unprecedented cooperation and collaboration. Cooperation and collaboration at the national level, at the international level, between the vaccine developers and the regulators, who would be the people who would give the approval for the vaccines to be used, even between developers of vaccines, and creation of joint ventures, which we've taken advantage of as far as things like manufacture are concerned. So cooperation and collaboration is one of the key elements that has come out from the pandemic. And the BMJ and ADB have both given you examples of what they've done to help with that cooperation and collaboration. And I would like again to pay my respects to the BMJ for the work that they've done, making the information flow related to COVID-19 manageable for all of us out there that are trying to deal with the disease. The other unprecedented approach that was adopted, which is incredibly important, was that there was an adoption of cost risk. Most vaccine development work proceeds in a series of stages that I'll describe in a minute. Each stage has a milestone or an end state, and at that point, the risk of continuing versus the risk of results so far were looked at mainly in terms of cost benefit. For COVID-19, the cost risk was adopted by governments, by organizations that came together to deal with the threat, and as a result, vaccines could be developed that even if they failed, the full cost of development would be covered. And so people were able to take the risk on of developing vaccines, even if they thought they may not work, in order that we had a large panel of vaccines. And incredibly, a number of those have made their way into use across the globe now. And we have a number of vaccines that we can use. Just briefly again, vaccination, for those that are not familiar, I think most people are, but for those that are not, it's stimulation of one or more components of the adaptive immune system and development of a memory response that targets the pathogen. We still don't know what a protective immune response is for COVID-19. We know some of the markers that indicate that an immune response has occurred, and we know that some of the responses to the natural disease should give us a marker for potential protection, but we still don't have defined the precise components of the immune system that we need to stimulate to give long-lived and extensive protection against Sars CoV-2. 2 points that I think are very important as far as this is concerned is that the primary function of the vaccination program is to protect the individual who's vaccinated. This is all about protecting individuals. Safety and efficacy is measured in terms of individuals. There is a secondary benefit that in the use of the vaccines amongst a large number of individuals, you can protect the population by limiting disease spread and transmission. And we're seeing that in countries that have achieved higher levels of vaccination amongst their population. But it's important that in terms of safety and efficacy, but especially safety, vaccine safety systems ensure that the vaccines are as safe as possible for each individual. And that safety extends through the manufacture of the product to ensure that the manufacturing produces a consistent product under manufacture that is safe, through the clinical trials, which I'll talk about, and also into the post-authorization and safety studies after licensure, which Dan will talk about, and I'm not going to cover here. Again, very briefly, I've put up here a comparison between the traditional vaccine development and the development with Sars Co V2. And what you can see at the top is that traditional development may take 15 years or longer, and it's clearly a sequenced process that leads from design and exploratory work all the way through to large-scale production and distribution. And if we look at what happened with the majority of Sars Co V2 vaccine development, and they're all different, but they're all very much similar, the process began with taking an approach that allowed for parallel versus batch processing. So as a vaccine developer in the past, I would develop my information in the lab. I would then submit that to a regulatory authority. That might be several 100,000 pages of documentation that I have to collect. And then I would submit it to a regulatory authority and they would review it. And then they'd come back and we would discuss a phase 1 trial, and we'd submit the documentation for the trial and then the results of the trial. And then if we approve, we go through to phase 2 and then phase 3. And then we would submit the license application. And in addition, at that stage, we would be looking at large scale production and distribution. We wouldn't do that early on because of the risk of producing something that isn't then licensed by the authorities. With SARS and COVID-V2, what was done was a lot of parallel processing. So a lot of the phases overlap. And I'm going to speak particularly about the overlapping clinical phases for phase 1 and phase 2. Dan will speak about what's happening in phase 3. I'll also briefly talk about the production, which is done at risk. But that assumption of cost risks for production failure, the collaborative rolling wave of regulatory reviews, and the use of emergency use authorization for the drugs versus full product licensure means that all of the things that are done to ensure the safety of a vaccine were done as far as these vaccines were concerned. They were just done in a compressed timescale. And where people have been hesitant about these vaccines because they've been developed very fast, the truth is they're very fast, but they've been developed using exactly the same approach, just in a slightly different configuration. So a quick run through some of the issues associated with clinical trials. First off, all the vaccines for use in humans are subject to review by regulatory authorities. Those safety reviews include vaccine manufacturing standards for purity, potency, and stability, and the clinical studies involving thousands of volunteer participants are conducted to assess the vaccine safety and efficacy. The initial clinical trials involve the monitoring and follow-up of volunteers for vaccine-related issues. These can be as simple as soreness at the injection site through mild, moderate to severe side effects. And if efficacy is often assessed in trials involving volunteers receiving the vaccine or a placebo, and in the case of COVID-19, both the volunteer groups and the placebo group and the vaccine group are monitored as far as the virus is concerned, and the development of COVID-19 disease. The immediate approvals, the approvals that we're operating under right now, were limited to emergency use authorizations. Those permit the widespread use of the vaccines whilst additional data was collected, the data that Dan will speak about. The first vaccine application for full licensure was made to the US FDA by Pfizer just 2 weeks ago on May the 7th. And they will be submitting both their phase 1, phase 2, and now their follow-up data as far as the use of that vaccine is concerned, looking at safety and efficacy in a large population. The second thing that's important about COVID clinical trials is that the types of vaccine that we were using and the technologies that were being used were proven vaccine platforms. And I've just put up here a table of the 4 most common vaccine types that are being used for COVID-19: the mRNA vaccine, the adenovirus vaccines, the subunit vaccines, and the inactivated vaccines. And as you can see, whilst mRNA is relatively new, with about 10 to 12 years worth of use, the inactivated vaccines go all the way back to 1890. So we're talking about vaccine platforms that are established. We know how they perform for the most part in humans. We know how they work, and we're able to leverage that in order to look at what happened for COVID-19. The focus areas for those clinical trials: phase 1 is immunogenicity, so an immune response of some sort, which of course, as I said, for COVID-19, we're still not sure what that is that provides protection, but we knew what we were looking for in terms of at least immune responses. Some early looking at dosing, at batch consistency, at tolerance for the vaccine and safety. And then in phase 2, efficacy and safety again. So safety remains consistent all the way through the trials. And the phase 1 and 2 clinical trials recruit volunteers and use inclusion criteria for enrollment. And what we're looking for during a dose escalation and a vaccine-related safety study is results that report any of the following 5. So these are what clinical trials focus on: adverse events, life-threatening adverse events or life-threatening suspected adverse reactions, serious adverse events or suspected adverse reactions, suspected adverse reactions, and unexpected adverse events. And if we look at the first of the published results, I picked this one not because it's a particularly important one, other than it was the first set of phase 1-2 results that was published. It's for the Pfizer mRNA vaccine. And as you can see, there were some 43,500 participants in this trial. It took place between July and November of 2020. The efficacy studies, because it needed to be studied in areas where there was active COVID-19 spread across 6 countries, there were 152 sites who were involved in the study, and there was a minimum of 2 months follow-up after dose 2 before these results were published. And again, looking at COVID-19, there were 3 things that were identified early on for this disease with regard to risk factors. The first was age, and so 42% of the people in this trial were over 55 years of age. Obesity, 35% of the people in this trial had a BMI of over 30, and pre-existing comorbidities or coexisting conditions, and 21% of the people in this trial had a coexisting condition. Those are all quite high numbers. Normal phase 1, phase 2 would not have a profile that looked like that. They would be a younger population, they would probably not be obese, and they are unlikely to have coexisting conditions. But it was felt that at this stage, those were important risk factors that needed to be looked at for the safety of the vaccine along with the efficacy of the vaccine. And I've put the results up here not for any particular reason other than to show you that they're classified mild, moderate, severe, or grade 4, which we'll come to in a minute. And they're looking at pain at the injection site, redness, swelling, the possibility of fever, fatigue, headache. Well, you can read it, it's all up there. But you can see these are not significant adverse events as far as the usual monitoring is concerned. What we're also looking for in these clinical trials is the possibility of a significant event. So the trial protocol, as far as safety is concerned, ensures safety and meets the stated objective, begins with a dose escalation scheme. So you start with low dose and go higher with the right number of subjects. And then you have a monitoring scheme. And what was important is that there is a high frequency of monitoring for observation, a good set of laboratory follow-up, and safety stopping rules that would lead to pause and halt if there were adverse reactions. And as far as these vaccines were concerned, the AstraZeneca vaccine, the J&J vaccine, which we'll talk about a little later on, all had pause associated with them. In the case of AstraZeneca and J&J initially, it was a single case of adverse reaction or unexplained illness. And of course, with the thrombolytic bleeding thing, it was associated with 8 vaccines. And Martin Postma, who's part of the UK Joint Committee on Vaccination and Immunization, said that regarding the issues with, and he picked out the J&J COVID-19 vaccine and the thrombosis issue rather, statistically, 1,000 COVID-19 deaths would be prevented for every 1 million people who are inoculated, while scientists would expect just 1 fatal case of vaccine-linked blood clotting. So the possibility of being exposed to COVID-19 and dying was much, much higher statistically than the case of blood clotting. But as Martin went on to say, but people don't think in terms of risk benefit. We give these vaccines to healthy people, so there's zero tolerance when it comes to side effects. So I leave you with a few predictions. It's hard to make predictions, especially about the future, but I would highlight in this just the fact that the vaccines will help prevent severe and moderate COVID-19 and reduce hospitalizations. That's absolutely clear. Equally, the vaccines will need to be modified to account for resistant variants and strains, and we're going to see that. So for the immediate future, we may need to receive additional booster vaccines, and it's possible, like influenza, COVID-19 may require annual vaccinations. And I'll leave it there and turn it over to Kieran and to the rest of the panelists, and I thank you for your time.
Thanks very much, Steve. That was absolutely fantastic. Now, before we move on to our next speaker, we have our second poll. So, Mark, if you can launch poll 2, please. Thank you. And Mark, if you can show us the results of poll 2, please. So that's very interesting. The majority feel confident, but still a significant number feel they need more support. So let's now move on to our next speaker. It's my pleasure to introduce Dr. Daniel Salmon. Dr. Salmon is Director of the Institute for Vaccine Safety at Johns Hopkins University, and his training, research, and practice interests in epidemiology and health policy. He is focused on post-licensure vaccine safety and the factors associated with parental decisions to vaccinate or not vaccinate their children. Dr. Salmon served as the Director of Vaccine Safety at the National Vaccine Programme Office, where he was responsible for overseeing and coordinating federal vaccine safety activities. And Dr. Salmon will be speaking about post-authorization safety assessment methods. Over to you, Dr. Salmon.
Well, thank you to the organizers for the opportunity to speak today. Give me just 1 second to get my slides up and running. So I'm going to talk today about post-authorization safety monitoring and pick up a little bit from the last speaker. So let me start with why monitor vaccine safety. And the overall goal is to ensure that the benefits of the vaccine outweigh the risks, both for individuals and for the populations being recommended the vaccines. And as was mentioned by that quote from a JCVI member, they're very high standards for vaccine safety for many reasons, but largely because we're giving them to healthy people. And our goal is to prevent disease, but we don't know with certainty who will be infected and who would have serious disease. Additionally, because we're vaccinating very large numbers of people, even a small relative risk can have a large attributable risk. And I'll give an example of that. Additionally, we want to optimize the use of limited resources. And importantly, we want to improve and maintain public confidence in vaccines. And ultimately, our goal for most vaccine preventable diseases, and certainly for COVID, is to reach and maintain very high levels of vaccine coverage in order to control the disease. So one of the challenges we face in vaccine safety, and I think this actually addresses one of the comments we received so far, but this concept, this fallacy that because one thing happens after another, it must be caused by it. And this is a human fallacy. This comes from our many years of history where we didn't have science, and people presume causation from temporality. One of the questions I saw that came in was, well, how come people are dying after they receive the vaccine? And, unfortunately, people die every day. So if we vaccinate large numbers of people, there will be people that die after the vaccine. And we have to be cautious not to presume causality from this temporal relationship. Adverse events following immunization, which mean exactly that, something that happens after the vaccine. This only says there's a temporal relationship. Well, this will occur. And it's important to differentiate between coincidental events and those that are caused by vaccines. So for example, in the US, we have 2,500 miscarriages and 3,000 heart attacks every day in the US. So if we were to vaccinate the US population one day, we would expect to see 3,000 heart attacks the next day simply by chance alone. So we need to anticipate these background rates of disease. And then we also need to rapidly identify and follow up signals so that we can determine, is this coincidental or is this causal? And unfortunately, robust science takes time and follow-up takes time, whereas public concerns and rumors can spread globally in seconds. So I was asked to pick up where the clinical trials left off. And let's start with the pros of clinical trials. They are the gold standard. They're double-blind, they're randomized, and this has tremendous power in reducing confounding and reducing bias. And it's much easier to draw causal conclusions from clinical trials than from observation studies. There are strict inclusion and exclusion criteria, and this both reduces the risk to participants and also reduces confounding. And then clinical trials are incremental, and this allows us to minimize risk while optimizing the information obtained. So these are the overall benefits, the pros, or some of the pros, the biggest pros of clinical trials, but there's limitations as well. Because of these inclusion and exclusion criteria, you cannot evaluate adverse events in persons that were excluded from the studies or inadequately powered in the studies. And this might be people that have certain medications or concurrent medical conditions, and you can't evaluate delayed adverse events. Additionally, because of the sample size, and we see in some of the COVID trials that they're quite large, 40,000, 50,000 people, they're really large studies, but you still can't evaluate rare adverse events or adverse events that occur in subpopulations that aren't adequately studied. And I make this point in this slide here. This was adapted from a slide that Susan Ellenberg published 20 years ago. And let me just take a minute to walk you through this, but this is really exemplifies the power but limitations of clinical trials and the number of people that are particularly impacted or could be impacted if you miss something. So here we see rates and a percent versus a percent. So this would be the background rate versus the rate one wanted to detect. So in this first example, it would be a background rate of 0.1% or 1 in 1,000. And if one wanted a clinical trial to detect a doubling of that risk to 0.2% or 2 in 1,000, this would require a sample size of about 50,000 people. With that said, if one were to miss this and vaccinate, in this example, the US population, this would all be with one dose, this could impact 200,000 people. So this really shows numerically, quantitatively, the limitations of clinical trials and, in fact, the number of people that can be impacted. Now, if you were to look at the number of people affected globally or in a country larger than the US, clearly the number would be much higher. Or if this risk happened after either dose of a 2-dose vaccine. So for this, we have post-authorization studies. And the goal here is to expand the safety profile of vaccines to include intermediate or rare adverse events, to look at subpopulations excluded from, inadequately powered in clinical trials, and to address emerging safety issues that can and often do arise. As a first step, and this is what many countries unfortunately rely on exclusively, are passive surveillance systems. And the purpose here is to detect a signal of an unanticipated event that may deserve follow-up. But there's a real limitation, which is that you cannot determine if the event is caused by the vaccine or coincidental in almost all cases. There are exceptions, but they're rare. And the reason for this is that you have a lack of good denominator data, and you don't have a good comparison or unvaccinated group. Additionally, you have a lot of potential biases, in particular, underreporting, incomplete reporting, and at times over-reporting. So in the US, we have a system called VAERS, the Vaccine Adverse Event Reporting System. It's co-administered by the CDC and FDA, and it accepts reports from anyone. Most reports come from individual patients or providers, but literally anybody can report anything. I could get an influenza vaccine and my dog got hit by a car, and I could make a report to VAERS, and it would show up in the database and become publicly available. The big limitations here are underreporting and incomplete reporting. And again, the goal here is to detect signals or to generate hypotheses, not to assess causality. And I really want to make this point because it's incredibly important. So if you think about a classic 2x2 table, classic epi 2x2 table, where you have the illness, yes or no, and vaccination, yes or no, VAERS, or a passive system, gives you cell A, and often a very poor measure of cell A because of underreporting. You don't have any information on people who weren't vaccinated, cells C or D, or people that didn't have the syndrome or illness and were vaccinated, cell B. So one needs to be very cautious in how they interpret data from these passive surveillance systems, and perception can be an issue as well. This is a quote from Barbara Loe Fisher. She runs an organization which many people have referred to as anti-vaccine. And well, I'll allow you to read it yourself. The point here is that it's very easy to misinterpret and overinterpret VAERS data. So it can be mis and overinterpreted, I think, intentionally, as is often the case. And sometimes people just don't understand what, in fact, passive systems can and cannot offer. To assess associations between vaccine and adverse events, most effectively, there's active surveillance systems. And we have a number of these systems in the US, which we use. The Vaccine Safety Data Link has been around for about 30 years. And this links large healthcare databases from a number of managed care environments. So you have inpatient, outpatient, pharmacy, laboratory data. And this has really been the backbone of vaccine safety in the US for many years. Many of the methods we use in vaccine safety come out of the Vaccine Safety Data Link. There's a number of other systems that have been used, PRISM for H1N1, though now it's focused on drug safety. CBER has a new system called BEST, and there's other databases like this globally. A large number of developed countries have large healthcare databases which are used to investigate signals that arise. And more recently, there was the development of the Global Vaccine Data Network, which brings together these large safety systems globally. And I really want to make the point that size matters. And this is an example from a hypothetical example. This is from modeling, but on the y-axis, you have the number of doses distributed in millions, and on the x-axis, the number of days before a threshold is met. And this is an example which shows, based on vaccine coverage, how large of the database one would need and how long it would take to detect a signal. And the point here is that size really makes a difference. And our next speaker is going to talk about vaccine confidence, but clearly we need quick answers to outstanding vaccine questions. And the larger the database for active surveillance, the quicker we're able to get answers. And I really want to emphasize that a signal is not the same thing as an association, which is not the same thing as causality. So one needs to be very, very cautious in making these jumps. So additionally, there's clinical immunization safety assessment centers in the US. It's called CISA. There's similar systems in Australia, in Canada. And the idea here is to study adverse events that are rare, which an individual clinician is unlikely to see. And these medical outliers, these rare adverse events, are very difficult to make advances on, similar to orphan diseases. So the solution has been the development of academic centers of excellence, which bring together vaccine safety clinicians to create a subspecialty with laboratory capacity. So I'm going to move on and just give an example which has caught a lot of news. This is the J&J vaccine, which was associated with TTS. And these slides are from Tom Shinkabahura from the recent Advisory Committee on Immunization Practices. And there was first the announcement of a pause in the J&J vaccine. And there were these cases that were incredibly unusual. This was looked at initially because of issues that were quite similar with the AstraZeneca vaccine. And again, this is an incredibly unusual clinical presentations. So the case findings for TTS, healthcare providers directly contacted the CDC about potential TTS cases. An FDA physician screens incoming VAERS reports daily to identify them, and then a further search was done of the VAERS database. The medical records were requested for all TTS case reports, and then medical officers at CDC and FDA reviewed these cases. There were 28 cases identified as of May 7th. The mean age was 40 years, the range 18 to 59. The onset from the time of vaccination to the symptoms was 9 days with a range of 3 to 15. All received the Janssen product. The majority were female. 19 of the 28 cases had CVST, none of whom were pregnant or postpartum. There was post-SARS infection in 5, and you see the risk factors here, which were really across the board. When taking these number of cases and breaking them down by age and by gender, and then looking at the reporting rate. So, you know, as I mentioned earlier, one needs very, very careful to try to get rates out of a passive system. So this is reporting rates. The CDC maintains data on vaccine administrations. So here we see the reporting rate was highest among females 30 to 39 and 40 to 49. But again, be very, very careful with this reporting rate, because as we know with passive systems, there is underreporting. How much underreporting? It really depends. It depends on whether or not people are aware of the association, how soon after the vaccine the adverse reaction occurs. It depends on how the patient presents and who they present to. For example, pediatricians are going to be more aware and more likely to make a VAERS report than an ER physician. We know, for example, looking at intussusception following the rotavirus vaccine, there was a nice study done, capture, recapture, that I found about half of them got reported to VAERS, but often it took a long time before those reports came in. So again, these are reporting rates. These are not actual rates of disease. When comparing this to the other two products that were used in the US, the Pfizer and Moderna, there was far more of these other products used and no confirmed cases of TTS reported to VAERS for either of these other two products. You know, and I'm sharing some data. This was a survey we had in the field at the time, very large survey of the public. You're looking at 3-day rolling averages of vaccine intentions. So in the dark green, you see people that had already reported being vaccinated. And as you would expect, this number increases over time as more people are vaccinated. I would really point your attention to the red line, people that reported they definitely would not get vaccinated. And when you look at the pause, when the pause was initiated, you know, you see what might be a small increase in the number of people that said they would definitely not get vaccinated. Likewise, when you look at people that said they would definitely get vaccinated as soon as possible, you see a decline in those number of people as well as people that said likely but not right away. So, you know, there's some indication that this pause may have had an impact on vaccine intent. So where are we at now? Well, the TTS following the J&J vaccine, very similar to those reported following the AstraZeneca COVID-19 vaccine. We need to increase our understanding. Who exactly are the populations at risk? What is the level of risk attributable to the vaccine? And how does that vary by subpopulations? For example, you know, men versus women in different age groups. And what is the biological mechanism? You know, when we see these very rare adverse reactions, often there may be a genomic factor. And then lastly, I would just say the potential implications here are far bigger for low and middle income countries. In the US, we had 2 other vaccines that were available. They were highly efficacious. We had fairly good supply of those vaccines. The decision to pause a vaccine such as this, when you have others available, is a very, very different decision than to pause a vaccine or not use a vaccine when there are no other alternatives. So let me stop there and thank you for the opportunity to share this with you today.
Thank you very much indeed, Dr. Salmon. Another fantastic presentation. Now, we have now time for our third and last poll. So Mark, if you can please launch the third poll. Thank you. And so if you can please take a second to answer this. And I think, Mark, we can just see the results for this one. So once again, a range, but lots of people saying no, not enough public information about safety, and majority saying the information not consistent. So really helpful. Thank you to all for participating. So now I'm delighted to introduce our third speaker, Dr. Priscilla Rupali. Dr. Rupali is Professor and Head of the Department of Infectious Diseases at the Christian Medical College in Vellore, South India. He was a Senior Infectious Disease Fellow at the Department of Adult Infectious Disease, Auckland City Hospital, and also runs an annual course in clinical tropical medicine at BMC. This provides lectures, bedside sessions, lab sessions by an internationally acclaimed faculty. Dr. Rupali will be sharing her experience as an infectious disease doctor in India, some of the questions she gets asked, and also about risked communication. So over to you, Dr. Rupali.
So hello everyone, and thank you for inviting me here. And thank you to the previous 2 speakers for their wonderful talks and for setting the stage for my talk. However, I have a disclaimer to make. I'm not a vaccine expert. I'm not a social scientist. Risk communication, my only qualification for this talk is the fact that we did some of it for the vaccine uptake in my hospital. So I'm an infectious disease physician. I was also in charge of the hospital infection control part of the COVID pandemic. And we did achieve quite a good vaccine uptake rates in our hospitals. So I'm just starting out with a disclaimer for now. So as you know, obviously I hail from a country where it's been really challenging. And we know over the last 16 months, this pandemic has ravaged the world and has caused many deaths and many cases. There have been measures which have slowed the spread, but vaccines hopefully will be our hope. So we're in the middle of our second surge, and it has decimated us and our country. We have seen many cases, 26 million is the latest, and we're counting. Death toll has crossed about 250,000. But there are good things is that we've also had many people who have recovered and gone home. So the problem for us in our country, coming from a low middle income country, is unfortunately it's not just a health crisis, but it's also an information and a socioeconomic crisis. And it is definitely going to create one of the deepest global recessions, and it's going to push many of our people into extreme poverty. So the surprising thing is many people are aware of preventive strategies. So why is that our vaccine uptake is so low? One thing is there is pandemic fatigue. People are not confident in their ability to control the virus. They don't have trust in the various state and governmental responses. And this pandemic is taking its toll on the fabric of our community structure. So what exactly is risk communication? And we were doing a little bit of research on it before we set out the stage for rolling out the vaccines in our institution. So this concept seems to have emerged from cognitive research in risk perception. So this is basically a lay concern regarding a hazard or a risk, which does not really correspond to an expert quantification. So rational decision-making is often clouded by biases or emotions. And of course, this worry and anxiety is amplified by the risk perception by the media. And these ups and downs of public distrust can actually lead to vaccine hesitancy. So what could be the impact of risk communication? So the most important thing will be behavior and willingness to follow public health and social measures can definitely be the weapons to stop the spread. But what can actually break the chain of transmission and mitigate the impact of the pandemic is actually risk communication. So why vaccination? Let's start with that. We know vaccines save lives, and vaccines save 5 lives a minute. And we know that the eradication of smallpox actually saved an estimated 5 million lives every year. If we had not eradicated smallpox, someone would have died from the disease every 6 seconds of every day. And we know that prior to measles vaccine implementation, which is as recent as 30 years ago, more than 6 million deaths occurred globally. So I think Professor Pryor has so beautifully alluded to the vaccine development process. But I'm going to dwell on something else. So what is it about this process that made people a little anxious? Funding was no obstacle. The whole world banded together. Thousands of scientists contributed to the effort. People signed up rapidly to participate in trials. These were tested in many participants in many countries and many other vaccines that we use now. Efficacy was able to be observed in real time in naturally occurring infections due to high prevalence all over the world. Pharma companies took financial risks, invested in manufacturing early on, and there was no delay in actually testing and rollout everywhere. So what exactly are the perceived issues in vaccine acceptance? So there are cultural and political differences in vaccine acceptance and hesitancy, and that is something that we have to accept and address. There may be a discriminated practice or discriminatory burden of COVID-19 in healthcare, and that is a perception that is widely available. There is COVID-19 vaccine hesitancy also due to some religious concerns. This may be also because of lack of engagement with the relevant communities. So when does vaccine hesitancy happen? Safety concerns heightened, especially when you're overestimating rare risk and underestimating common risks. There is over-reporting of negative information, especially in the media and with all the social media that is circulating today. Unfortunately, there is an overabundance of negative information. Sometimes actions are perceived more harmful than omission, and I can tell you how many people I had to talk out of not wanting to take the vaccine. There's also a risk ambiguity and freeloading. So people think, okay, if somebody else takes the vaccine and there is a high vaccination rate everywhere else, then maybe I don't have to expose myself to the risk of taking this vaccine. And the herd immunity is going to protect me and is going to protect my children. So what facilitates vaccine uptake, and what have we observed today? So although most people and their children get vaccinated against common diseases, there is definite variability between countries, cultures, demographic populations, and ethnic groups. But one thing I think is noted everywhere, that people are more likely to vaccinate when it's convenient, if it's free, if it's easy, if they have confidence in the safety of the vaccine and they trust the system that delivers it. But the most important, when their healthcare professionals recommend it. So what about the acceptance of healthcare professions? And that's where we worked tirelessly in our institution. So healthcare acceptance of COVID-19 vaccines is important. And that is actually borne out by influenza vaccination and the poor uptake by the healthcare workers in various communities. I can tell you there is very poor uptake even in the healthcare system that I work in. People know that vaccines can protect them, their families, their friends, their patients, and their community at large. So their recommendation is actually key to encourage others to vaccinate. So what are the variables that can increase hesitancy? One, there's very little research towards the factors that can contribute to vaccine hesitancy. And all of us need to do our bit to do research to examine factors that may lead to hesitancy toward the COVID-19 vaccine. One of my colleagues is actually closely working with a social scientist, and we hopefully will have data in a few weeks with regard to this. Some people do oppose the vaccine for ideological reasons, for some political reasons, there may be conspiracy theories, and of course the influence of anti-vaxxers as well. So how do we set the communication agenda? And UNICEF and WHO have absolutely excellent resources for this. We do know that after there is initial hesitation, this actually resolves as the program becomes established. Transparent and effective risk communication can of course assist with this process and smoothen this transition. The communicators must be aware of the various cultural and emotional differences and also be able to recognize the adversarial and the misinformed people and be able to address their concerns. So risk communication, of course, has to acknowledge side effects such as fever and muscle pain. Our previous speakers have alluded to the so-called rare side effects which got so much publicity in the media. Ironically, these side effects of fever and muscle pain should actually show that the vaccine is working because it's preparing the body to fight the disease. And that is very important to be able to communicate to the recipients of the vaccine. Important to engage the communities and most importantly, get the healthcare workers on your side so they can promote effective communication and vaccine safety. So what were the various myths that we encountered when we dealt with it in our institution? Of course, the usual ones: it modifies your DNA, it is not safe to take while you're menstruating, it gives you COVID, it was developed using fetal tissue, I've already had COVID, so I don't need the vaccine is expensive, and actually the vaccine was being rolled out freely. And once I'm vaccinated, of course, I don't really need to wear a mask. What about vaccination? Can it be made mandatory? And we don't really have a clear answer to that at this point in time. Can we legally compel people? And could we limit access to schools, services, and jobs? And it's unclear whether it is right now ethical to do so. So we have to accept that there is equipoise here. So I guess what are the principles? And this is what I think the principles could be. Consider political, social, and economic factors. You analyze the risk and risk perception. Most importantly, summarize the evidence and make it easy for them to follow and to arrive at an informed decision. Dialogue with the target population. You allow them to express their fears. You clarify their misconceptions. You may have to go back to them again and again, which is what we found. And effective communication should actually minimize resentment from those who feel excluded from policy making. So we did some of this in our institution. And so we started out with first announcements. We also have a clinical infectious disease society, and we used that as well to make announcements. We had a very active team. We had FAQ documents in different languages. We held various public meetings and conferences. We had continuous dialogue addressing anxieties and fears. The one thing we did was we enabled those who were vaccinated to become facilitators and encourage vaccination. So I come from an institution which is almost like a little kingdom, and it was challenging for us. We started working very early on before the vaccines even became available because we knew it was going to be a problem. We have about 12,000 employees, and each employee has 5 dependents, and we take care of the dependents as well. You can imagine it is a massive undertaking. So the first thing we did was sometime in September 2020, the government had asked us to send in the names of the healthcare workers to provide the vaccination. Initially, there was so much hesitation. So that actually, we had to work around, encourage people to first thing, give their names in. Then we formed a committee which for advocacy. So we had people, various experts coming into the committee who helped disseminate the knowledge with regard to the vaccine and its safety. The first thing we did was we introduced FAQs in English before the vaccines rolled out. We modified it after feedback. Then the first thing we did was that we did go about debunking certain myths and then provided certain facts. So first thing to make clear was the risk of the disease far outweighs the risk from the vaccines. Side effects are transient and increased serious adverse effects are exceedingly rare. The other thing we did was provide them information about various countries and how many of them were willing to be vaccinated. We did various talks. These were our FAQs from our Hospital Infection Control Committee. We had to roll it out in different languages. So as you know, there's a lot of diversity in India. So a lot of our healthcare workers come from different parts of the country. And so we have workers who are fluent in Telugu, Tamil, and Hindi. So we need to actually make FAQs in different languages as well. Circulating myths. This was a great challenge. So in fact, we had very educated doctors coming and asking us, okay, does it actually alter DNA? And I think that was close on the heels of the mRNA vaccines being rolled out from the United States. And then of course, is the vaccine going to make me infertile? That was one of the major things. Does the vaccine actually have a microchip? And there were constant messages from the TV and the media that were making people extremely anxious. There were other myths, and we called upon experts like Dr. Gagandeep Khan to give us various talks. And they also helped us debunk some of these myths. So were the vaccines rushed? Are they unsafe? And the ones that I mentioned before them. Adverse events, and I think this got a lot of publicity and close on the heels of Europe stopping vaccination, especially the Norwegian Institute of Public Health. And we were at that time rolling out the Indian version of the AstraZeneca vaccine, or Covishield as it's called. So we had a lot of anxious people calling us constantly. So what is the risk of blood clots and what should I do? And this as well, we had to debunk a lot of these queries. And as you can see here, Dr. Cherry or Dr. Kang helped us with these kind of information. So COVID infection, yes, the clots are more likely to infection than really it is with the vaccine. And we know that there are risks of blood clotting that can be seen with smoking or with birth control pills as well. So really, it's much lower than that. The third thing we did was lead by example. So you can see here that is my hospital infection control officer and our administrators. So on the first day when the vaccine was rolled out, they were the first ones to take the vaccine. So that is how they led by example. All the administrators, the director, the medical superintendent, members of the administration lined up, took the vaccine. So the first day when we rolled it out, we were prepared to get 500 people taking the vaccine, but we only vaccinated about 20. It became easier with time, and then eventually we went on to vaccinate we reached about 700 in a day. So it took time, but we got there eventually. We had people there constantly, so this was the area where they received people. These were people who were kind of registering them on the government portal, making it easier for them so that they were not getting frustrated. We had people sitting there waiting to receive the vaccination. We tried to make sure that there was social distancing everywhere. So we had doctors on constant call there. So you can imagine in a hospital with 12,000 employees, how many doctors needed to be on the floor to ensure that there were no serious adverse events. We had a checklist prior to receipt of COVID-19 vaccination. So there were lots of questions. So we had one person posted there just for that. So have you had anaphylaxis, and you're pregnant, you're breastfeeding. And at the end of it, we kind of incentivized it. We gave them kind of a vaccination card and said, okay, look, you're vaccinated. The government also did its bit, and it also provided us with this kind of a vaccination card. I hope I've provided you kind of a bird's eye view of what we did with regard to risk communication and how in our small way, in our small area, in our small part of the country, we were able to achieve actually 90% vaccine uptake, and the only 10% that did not take the vaccine was because of pregnancy or lactation or serious anaphylaxis due to other vaccination. So thank you for your attention, and look forward to the question and answer session.
Thank you very much, Dr. Pally. That was brilliant, and we have now 3 excellent presentations, which are impossible to sum up. But suffice to say, what I've learned is the concept of parallel processing from Dr. Pryor, the idea of causation, temporality, and association from Dr. Salmon, and the complexity of that. And what you started your talk with, I think, Dr. Pally, when you talk about an information crisis and a socioeconomic crisis as well, which I think is really important points to be made. So we've got lots of questions, and let's start with the first one. Are there any safety issues if 2 different types of COVID vaccines are taken for the first and second dose? That's a question that's come up in different formats lots of times. So who wants to take that one?
Yeah, I don't mind addressing that, and others might want to jump in. And I think that relates to a lot of the other questions. And the simple answer is, based on data, we don't know, because those trials haven't been done. So we got questions such as switching from one dose to the other, or questions about how long will protection last. And, we need the science to answer those questions. And because the trials haven't been done mixing different manufacturers, we don't really know the answer to that.
Thank you. Steve, do you want to add anything to that?
Yeah, firstly, agree entirely with Dan that the results of the studies have not yet been fully published, but the studies are being undertaken. And in fact, the first results of a study involving the combination of the Pfizer vaccine and the AstraZeneca vaccine were disclosed in Lancet on May the 12th. And they did a very interesting study where they did 2 doses of Pfizer or 2 doses of AstraZeneca, or Pfizer, then AstraZeneca, or AstraZeneca and then Pfizer. So they produced the safety results as far as that's concerned. And what they observed was that the use of homogeneous vaccines, so the same vaccine twice, resulted in lower reactogenicity and side effects than if you mix the vaccines. But even the mixed vaccines didn't result in significant adverse events. But that's the first data that's come out, and I'm happy to share that online afterwards. But it was published on May the 12th in a letter to Lancet.
Thank you. Let me read out another kind of follow-up question.
Can I just come in on that, please?
Please do, Patrick. Yeah.
Yes, this is a very big issue for most of our developing member countries, because the cheapest vaccine is AstraZeneca, and that's the vaccine that COVAX gave to many countries, and so the challenge is that countries did two approaches to deliver the vaccine. Some countries gave everybody the vaccine that they received with an understanding and anticipation that 8 weeks later the second dose would come. Other countries gave the doses to 50% of the population and kept the other 50% of the doses. And for countries that gave all the doses at once, so you have millions and millions of people out there who got only one dose. And right now, and since the bulk of the vaccines came from India, the AstraZeneca vaccine came from India, and they've just increased they've just indicated yesterday that the next dosages will only come from India in October. It means that there's going to be many people who have received the vaccine and have months to wait for the second dose. And a lot of people are saying they're not going to wait that long, and that they'll look for other alternatives. Over to you.
Thanks, Patrick. That's very helpful. A follow-up question related to that: if you do have another type of vaccine as your second dose, is there a safe interval we can recommend for the next type of vaccine dose? Anybody want to have a go at that?
Well, again, pointing to this limited study so far, they actually conducted those studies with a 28-day delay for the second dose, and also a 12-week delay for the second dose. So as Dan said, we're still waiting on results. Many of the things that we would like to know the answers to are going to require studies that are ongoing right now, including, as I've seen on the question and answer sessions, studies in younger children, in under 12s. Those studies are just beginning now, and it will take some time before we have enough people in those studies to be able to get the results out. It's important that people recognize gathering the data and putting out the data when there is sufficient to make it statistically valid is an important consideration. Putting results out early just to get results out there that aren't statistically valid doesn't really help us. It just creates more confusion. Thank you.
Thank you. So let's change tack now to a different subject: how to address mothers who are breastfeeding and want to take the vaccine. Should we advise them to continue feeding or stop feeding and then get the vaccine? Is there an evidence-based answer to this question?
Stephen may add more, or others, than I do. There's been, in these trials, there were women that were inadvertently pregnant or got pregnant in this study, and they were observed and there were no problems that were identified. There's been other passive surveillance that hasn't shown a problem around pregnancy or breastfeeding, but to my knowledge, there haven't yet been clinical trials among pregnant women that have been reported. So, there's some evidence. The US recommendation is that in consult with the clinician, a decision is made. It's not a contraindication to vaccination.
I might come in, Kieran. What we did in our institution was anybody who was exclusively breastfeeding, we said no, but if they were willing to take the vaccine, we actually allowed them to take it if they were beyond 6 months. So that's what we did, though initially there was some hesitation because The way the vaccine portal is designed is doesn't allow breastfeeding mothers to be registered onto the portal. So we kind of explained it to them, explained all the risks, and if they were willing to stop breastfeeding, especially if they were beyond a year, we would tell them just stop breastfeeding, but take the vaccine. So that's what we did, but we left it to the healthcare workers to decide beyond 6 months of breastfeeding.
Okay, thank you. Sorry, Steve, yes.
Now, just to follow up on what Dan was saying, I am only aware of one study that's been done in a cohort of 84 individuals who were breastfeeding and were immunized with the Pfizer vaccine. And there was no apparent problems associated with that. But there is, in fact, a fact sheet that's been put out by the US National Institutes of Health related to use during lactation, and we can make that available to people.
Okay, thank you. Next question. Thrombotic thrombocytopenic purpura has been reported with the AstraZeneca vaccine. Have there been reports of thrombotic episodes without thrombocytopenia? And if yes, is that a contraindication for the second dose?
So I'm sure that there's been reports. I don't have the data in front of me, but as I said, there would, bad things happen to people all the time, and thrombotic events happen to people with some frequency. So simply because there's reports of that relationship, one can't presume an association. I'm not aware of a contraindication for a second dose. But I think that's something that would be under the consult of the clinician.
Okay, thank you. Next question relates to a scenario where a person gets their first dose of vaccine, then has COVID infection. Should the second dose of vaccine be delayed? Who wants to take that one?
Well, I'll answer on the basis that it's not been studied in any clinical trial. So on that basis, there is anecdotal evidence from people as far as that's concerned. The evidence that's being collected at the moment that is most linked to that is studies that are called vaccine breakthrough events, which are when we know that as a result of vaccination, there hasn't been full protection from COVID. And that's a fairly rare event. It's about 1 in 10,000 people who are receiving the vaccine might end up with a COVID event. Of those, only about 1% actually have a significant COVID disease associated with it. So it's a very rare event. And as far as I'm aware, there is at the moment nothing other than the guidance of an individual health practitioner to say that once you've had the first dose, you shouldn't go ahead and have the second dose.
Yeah, I would just add that a lot of these questions are really great questions and of clinical importance, and they're going to come from your patients. And at the end of the day, we're going to be dependent for much of them on real world data. I mean, some of these questions will be addressed through clinical trials, but those trials are expensive and take a long time. And if one tried to answer all of the relevant questions through clinical trials, it would just take exceedingly long. So it takes time in a real world setting to do observational studies. And frankly, they're more difficult to do because they're imperfect, because we don't get to randomize people. But that's how we end up getting answers to many of these questions.
Thank you. I agree. They're great questions. They're also quite difficult questions as well. Let's move on to another difficult question related to that one. Say if you have the first dose of vaccine and then you catch COVID, is there an extra chance of a hyperimmune response to the second vaccine? Do we know?
I have no data.
Yeah, I don't think this has been studied. I mean, someone could try to answer that question biologically. I'm going to pass on that one. But what we don't have is data to answer it.
Okay, thank you. A very straight question here. Does the Pfizer vaccine protect against the Indian variants?
again, real world data that's being collected right now. Lab data suggests that it does, and that it is at least partially and probably effective, but real world data, we'll have to collect that, and we will know sometime down the road. Again, going back to the point I made with the silly slide right at the start, we are literally building this aircraft as we're flying, and some of the parts that we would like to have are not yet there. Some of the other parts, the essential parts, we have got, so we can actually fly it, but we're missing some of the bits we would really like to have.
Thank you. Another question, public health type question. With the scarcity of vaccines for low-income countries, can the available vaccines for those countries be used in areas where the infection rate is high? So if there's a particularly high infection rate in a sub-district, can that area be placed under lockdown with mass vaccination? Dr. Rapalli, do you want to take that one?
So I guess that would be an appropriate thing to do. I guess if you have a high.
Sorry, Dr. Rapalli, I think we've lost you there temporarily.
While Priscilla's waiting to come back here, and I'll pick up on it if I may. That approach is being used, and in fact it's being used where Kieran is now in the United Kingdom. So there are efforts to run very, very high levels of vaccination in areas where there are considered to be higher risks from the disease right now, in particular with at least one of the variants that is currently circulating. And so yes, that approach that was described is being used. It's very intensive in terms of vaccines, it's very intensive in terms of logistics, but it has been used.
Okay, thank you. Another question. How can we know whether the immunity after COVID vaccination is adequate or not in an individual?
We can't. And in fact, it speaks to what it's the inverse of what Dan has been speaking about, is that right now we are largely taking our effectiveness based upon the clinical trials and then the extensive use of these vaccines in the population. It's the fact that as of now, we don't even know what an effective dose is for a population or an individual. We don't know the most likely route of transmission. We don't know what the immune response is in terms of the innate response, the humoral response, or the T cell response. We know very little as of now about the response of an individual, and therefore what an immune correlate or a level of immune correlate needs to be to provide protection. Those are all studies that will be retrospectively filled in as we go forward. What we do know is that the vaccines have been very effective where they've been used in reducing the severity of the disease, and we are seeing reduced transmission in areas where vaccination has been high. But in terms of the specific immune response, we know some of the results in terms of neutralization antibodies and humoral antibodies. We know T cell responses, but what's protective? And what do we need to promote for long-term protection, and how long that protection will last? We don't have that data at this stage.
I would add, there's so many great questions coming in, and I think the principle, whether it be a health authority speaking to the general public or a clinician talking to a patient, is this is what we know, this is what we don't know. This is what we're doing to figure out what we don't know, and this is what you should do for now. And if we take that approach, we're far more likely to have people trust us and to be able to make changes to our recommendations without the perception that we don't know what we're doing. The truth is, this is an emerging infectious disease. There's an awful lot we still don't know. We're very fortunate to have amazing tools as these vaccines that we're seeing. I mean, it's really remarkable. But if we're forthcoming in what we know, what we don't know, what we're doing to fill those gaps, and be clear on what the recommendations are at this point, I think that's the best way to address these very important and relevant questions.
Thank you. Absolutely. Let me ask you another question, which I think is maybe a little bit more straightforward, but an important one. After a non-vaccinated person gets COVID and then recovers from COVID, is there an indication to get vaccinated? And if yes, after how many days after the COVID infection?
So, I mean, the short answer is yes, because we don't know how long COVID infection will provide protection. I think that the recommendations and how long to wait are varied by country. I don't think that there's a single number that's been agreed upon globally. And, you know, it's not based on randomized controlled trials, right? I don't know if Stephen wants to add to that.
No, I think you're absolutely right. You know, it would be nice to have the data, but we don't at this stage.
Okay, thank you. Another question. After completing 2 doses of vaccination, can a woman get pregnant or should she wait? Or is there any other criteria that she should use before making that decision?
I'm quite confident that she could get pregnant after the vaccine if she could get pregnant before the vaccine. But we don't have data on this. I'm not aware of any recommendations in this regard.
I agree.
Okay, thank you.
I think Priscilla may be back, Kieran.
Oh, fantastic. Great. Priscilla, hi.
I'm sorry, there was some issue with my internet. So.
Okay, great to have you back again. Next question. Is there any vaccine that can be given to a child under 10 years?
That trial is ongoing right now. In fact, children under the age of 5 are currently being administered the Pfizer vaccine in the United States of America in a clinical study. So at the moment, as far as I'm aware, there is no age limit, upper or lower, that hasn't participated in at least some part of a trial, although again, those are in the very early stages.
Thank you. I've got a very straight question here. I don't know, is there a straight answer to it? What is the duration for which the AstraZeneca vaccine remains effective?
I could argue that you need to define effectiveness before you can answer that question, but the answer is we don't know. We simply don't know. Those are real-time data that we're going to collect. How long does a vaccine last? When do we need boosters? What boosters are needed? They're all exactly as Dan said. We can only tell you what we can tell you. We don't know. What we do know is that from previous studies using similar types of vaccine to the AstraZeneca vaccine, so the adenovirus vaccine platform, the protection offered associated with SARS and MERS when we take a vaccinated person and put that material into an animal and then challenge the animal, they're actually quite long-term in terms of the protection. But whether that translates into protection for an individual who becomes exposed to the pathogen at a later date, we don't know. Again, those studies are obviously unethical and will not be done. But in a population, you would need a large number who have been potentially exposed, which goes back to that first study that I showed. You needed 6 countries in order to find enough people with enough circulating COVID to be able to show efficacy.
And in time, we'll probably come up with a good correlative protection, and that may be how we answer this question at first, at least.
Okay, thank you, and thanks to everybody who's submitted questions. And we've got limited time, and I don't think we're going to get around to answering all of them. But before we end, I'd like to ask each of our 3 panelists for one final reflection or key learning point for our viewers, or one question maybe that you saw that we haven't answered thus far. Dr. Rupali, can we start with yourself, please?
So thank you, Kiran. And I guess my key message to everyone would be, yes, we are uncertain about the durability of immunity after the vaccines, whichever vaccine you're taking. So whether it's the AstraZeneca or the Covishield part, the variant of the AstraZeneca vaccine, or whether it's the Indian Covaxin, which are the two vaccines that are available right now. But I think we should encourage everyone to take it because the side effects are very few, and we need to kind of encourage each other to take it to bring all the mortality and morbidity down. So that would be my key message, really.
Thank you. Thank you. Dan, any final point from yourself?
Well, there were a bunch of questions about the AstraZeneca vaccine and the J&J vaccine and blood clots, and should it be recommended or given to women or people of a certain age? And I would just remind people that this is all about risks and benefits. And in safety, we're interested in, we study rare adverse reactions, and as I try to articulate, they're very important. But at the end of the day, it's about risks and benefits. And if that's the vaccine that's available and the benefits clearly outweigh the risks, which have been the conclusions of European and US regulatory authorities, then the decision should be based on that risk-benefit calculation.
Thank you. Steve, any final point or point from yourself?
Yeah, thank you. I agree. I think the questions have been great. And I think they point to an issue that Dan has spoken about, topic that Dan has spoken about. We began with an enormous amount of uncertainty about COVID-19 and SARS coronavirus 2. We've answered some questions. It is remarkable that we sit 15 months from the real first wave of this disease with vaccines that have been used on a global basis. So we have some certainty, but we have a lot of remaining uncertainty. And unfortunately, that's going to remain with us for some time. Hopefully, some of the uncertainty remains long after the disease has been pushed back by the vaccines. But I agree with Dan, the vaccines have been a remarkable outcome as far as this thing is concerned. And we hope for more of the same.
Thank you. And Patrick, I'll just give you the opportunity, if you wish, if you have any final comments or reflections before I then wrap up.
Excellent session. Some of the questions that I've received from many of our member countries, many of the experts from community level have been responded to, where we have the information and also where we do not have information. So thank you very much to our esteemed panelists.
Thank you, Patrick, and thanks very much to all our panelists. We hope you found the presentations and discussions enlightening and helpful. I'd also like to thank the Asian Development Bank for their partnership. And finally, thanks to all of you for participating. We really hope you found it valuable. Don't forget to register for the COVID-19 Information Centre to get the latest evidence-based guidance and e-learning from BMJ on COVID-19 and its comorbidities. A recording of this webinar will also be made available in a week's time, and we'll put that up on the site. Please do help us by filling in our feedback form. Lalitha will add the link in the chat box. If you require a certificate of attendance, you'll have to fill out the form as well. So please do that and we'll get back to you. So once again, thanks very much to our panelists, and thanks very much to all of you for taking part. Have a great rest of day or evening, wherever you are. Thank you.
Thanks, everyone. Bye now. We staying briefly.
Everyone. Bye.
Bye-bye.